Good morning, everybody. Thanks for coming. I'm McLaughlin. I'm from the Biophysics Facility here, and today we're going to talk about data fitting, errors, and binding curves. I'll start with this quote from Richard Henderson, and which really shows us that we can't underestimate the ways that we can invent new ways to deceive ourselves. And in this lecture, we'll see which ways you could possibly deceive yourself with bad data. Here's a first example here. It's a functional MRI of a salmon being shown pictures of human emotion, and they're looking at brain activity. And you can see these red spots here. Wow, that looks fantastic. Very, very interesting that salmon could actually recognize human emotion, but there's one problem, and that is the salmon was dead. And the reason for this is because they didn't have a good background correction. And the question you always have to ask yourself during all your research, your PhD, is, are you looking at a dead salmon? So today what we're going to do is we're going to have the beauty of mathematics, and we're going to try and fit your data. And hopefully we're going to end up in the bottom right-hand corner here where you've got beautiful data and you've got a beautiful mathematical model. But let's first of all think about what we're all doing. So initially, you've got this biological model of what you think is going on. You're going to have some methods and some controls. And then you should ask yourself, well, is it reproducible? Is the target you're actually looking at to interpret this biological model actually valid? And this is going to give you a physical model, which is then going to hopefully give you ideas about the biological model. But there's a bit of a crisis in science at the minute, especially in pharma, where lots of drug trials have actually failed at the first level. And that's because about whether the science is actually reproducible. And so Bayer Health and Amgen and this cancer biology project have gone back and looked at published targets. Bayer Health could only find that they could only validate 25% of the targets. Amgen, they could only confirm 11%. And in the cancer biology, only 45% success rate of 112 studies. So the issue then is, of course, they're basing all their data on the literature. They're going after these drug targets, which are supposed to be controlled with biological model, but the targets aren't actually valid. Also, journals now expect that you have to make things reproducible. They want clarity on all your data. And lots of times now, they actually expect you to deposit your raw data. So what are the roadblocks to reproducibility? Well, first of all, there's kind of a pressure to publish. You must publish, and that could lead to kind of cutting corners and really blinking yourself. And this blinking is called cognitive bias. One thing could be confirmation bias. This is what the field says. This is what your supervisor says. This is what should happen. So you're looking for data that can confirm what you think is happening already. You could have selection bias, where you've got a whole bunch of data, but you only select the positive ones, forget about the negative ones. And so really, you're just kind of forgetting about the bad data. And so you've got selection bias there. There could be a bandwagon. In a field, sometimes they're driven by, this is the mechanism, this is the way things go. And I mean, a case in point is if you want to go and read up about Alzheimer's research and how a part of that field in terms of antibodies development was against a target that wasn't valid, and has wasted a lot of time and a lot of money. Sometimes you can get a cluster illusion. We're very good as humans at seeing patterns. It's just random kind of data. One example was that back in kind of the 19th century, people would look up at Mars and imagine there's actually canals. But that was just joining the actual dots. And then there can be a reporting bias as well. And this is particularly prevalent in the literature. It's very difficult to publish a negative study. I tried this, it didn't work. It's always about positive results, So again, there could be a bias there to kind of thinking, well, I can only publish, positive stuff. And also, as you can see, when you come to read the literature as well, often there's obviously poor methodology. At the end of a paper, it's a very, very small kind of paragraph about what the method is. Sometimes things are left out. Sometimes very, very crucial things are left out. And that's, maybe that's why, you can't reproduce it, because you don't have that buffer or that component, or you didn't do it at a certain temperature. And also there could be a lack of statistical power. You'll often see, especially in the paper, in the news, they will announce big medical breakthrough or whatever. And then if you look at the study, it'll say, oh, they studied 12 people. So is that valid or is that not? And the answer is no. Okay, so let's think about what your experiment will tell you. You want to get this kind of physical model. You're going to have some kind of signal. You're going to ask what the assumptions are in that signal. Is the signal representative? You're going to have raw data. Is that raw data accurate, precise? We'll look into that. What are your controls? Are they good enough? How robust is your assay? You're going to have corrected data. Are you looking at the right time, the right concentration range? You're going to have some kind of data model. You're going to impose upon your data, and you're going to fit that data, and hopefully that's going to give you validate your physical model, and then that's going to give you insights into the biological model. So here we're going to have examples of things that can go wrong. So in a case in point here is the physical model. I've got an enzyme that's going to bind a substrate that then is going to be turned over to a product. The product is then going to leave. And I'm going to be looking at the KD here, which is just then the ratio of the reverse rate over the forward rate. And in this example here, I was looking at an ATP that bound ATP. And when I bind into the active site, you've got a change in the intrinsic protein fluorescence. And I could then follow that binding then by this change in fluorescence. And here you're using against non-hydrolyzable ATP, PPMP, which I'm then going to put a data model onto. And I'm going to make assumptions that it's a one-to-one binding, and that the concentration of the complex formation here, ES, is then proportional directly to the change in fluorescence, because I'm going to say the catalytic step here, K3, is going to equal to 0. I'm using a non-hydrolyzable ATP analog, and so therefore I don't expect the enzyme to be able to cleave that. But what if the physical model is incomplete? What if, say, there's an extra step? So the change in fluorescence is then equal to this conformational change in, say, the enzyme, or perhaps then there's an inactive enzyme. The enzyme itself is in two kind of states, binding and non-binding. So you really have to ask yourself, can the physical model then be ascribed to a specific step in the reaction? And are there any missing steps? A good question here is the signal representative? So this is an assay I was developing. We're developing a tool compound where we're getting to look at drug binding to an inactive kinase. And so we had a reporter group that we knew bound into the active site of the kinase, and then that was directly coupled to a fluorescence molecule here. So you can see here at the top, this is the reporter molecule here that should bind into the active site. And so then as you titrate the protein increase, that should increase the polarization of this little molecule, and we can get a binding curve. Okay, that looked all very, very good. But then when we did the control, where we just titrated in the fluorescein, which is the fluorescence group, we got exactly the same KD. So hence the binding is non-specific. Well, how can you actually check that you've got a specific binding, especially when you're using kind of large hydrophobic groups that are involved with these fluorophores? Well, one way to do is if you've got a test compound that's a positive control, where you can actually displace, here is binding displace the active site inhibitor. So in this case here, you've got 3 different probes here, they're all fluorescently labeled. And then if we then try and displace them then with a known, in this case, a kinase, a universal kind of binder, staurosporine, we can see that in green, only one of them truly displaces, and the other one you're getting partial. So we can say the one in red and the one in blue, they're not really binding in the active site at all. So the question you always ask yourself, what are your controls and are they good enough? So what we want to do ideally is we want to find this true parameter. So this is in the cyan in this middle of this bullseye, but in reality, things aren't as nice as hitting the target all the time, where you get a very, very precise and a very, very accurate readings. Often what we will get is that there will be some distribution of the recorded value. And what we have to do then is actually, average more, take more data, so that then we can actually get that the population, and then get the average, get the mean value to get the true representation. Of course, oftentimes you might get this kind of case in the top right corner, where you've got a very, very accurate reading, but it's not very precise. So this can be useful in saying, things if you just want to see differences, like differential scanning fluorescence. There's assumptions in there that it's a true equilibrium, and which may not be because of aggregation, but you can get a very accurate value. And if you're just looking at, say, drug binding, where you're going to get a shift in the TM, it doesn't really matter. You've actually got the actual true thermodynamic TM. It's just a matter of being able to see that distance between the two values. So you need very, very precise. So what things can lead to inaccuracy? You could be random errors, such as we call that noise, and that measure of that as a signal magnitude relative to the noise. It could be the position of the tools you've used to prepare the sample. Your Gilsons are slightly off. Processes may be stochastic, as well as deterministic. So it could just be that, yeah, it's just going to be random, how things bind. So these random errors and the selective precision, these will all average out. The more you do, then it should average out. If you've got systematic errors, which we call mistakes, then that could be due to like instrument calibration. You've got the wrong data range. You've got the photo multiplier too high, so that you're saturating the signal, or it's too low, and so you've got very, very hardly any signal at all relative to the noise. It could be that the calibration of the tools, in kind of use in the preparation. So imagine that your weighing balance is off, or you haven't made the buffer up in the right volume or something. Temperature calibration is something that we tend to forget about, but obviously everything is a reaction, and that's really dependent upon temperature. And again, we may not leave things long enough for calibration. So these systematic errors or mistakes, they're not going to be removed by accuracy. No. Let's go back. These systematic errors or lack of accuracy are not going to be removed by the averaging. So how can we get better measurements? Well, in the components, make sure your buffers and are filtered. Take care of aggregation. Check at the end of the reaction. Make sure everything hasn't aggregated. Make sure the concentrations are accurate. How do you measure it? A lot of people do nanodrop. That necessarily may not be the best way, because you're not taking into account any light scattering due to aggregation. So take UV specs or AAA analysis. Is your sample stable? Have you got the right pH, temperature, the time? Are all your buffer components needed? This is something we come across quite often in the biophysics facility, where people will have buffers that are just the lab buffers because they are the magic buffers. And so if you ask people then, well, why do you have those components in there? Well, it's kind of like, well, this is the lab buffer. This is, my supervisor has told me to use this. And it may be that actually for your system that you're working on is slightly different to what the lab's been working on previously. And you may not need those components, or you may be needing to change the conditions. Is your buffer stable? If you have components like DTT, they will oxidize over the time course. Buffer components can go off as well. So what kind of methodology to use? Have you got a good signal change? Have you considered the background? What are the effects of the buffer itself? Are you doing accurate titrations? What's the temperature control like? Have you let enough time for equilibrium? We'll see this in a minute or two. And what are your controls? It's always good to have a positive control if possible, and also a negative control as well, so you can kind of see how robust your assay actually is. So in talking about robustness, this tends to be something that's more done in pharma when you're doing high frequency screens. This is an example here, because you're going to be screening, thousands, if not a million compounds, and you need to ensure that you're actually going to get a good hit rate. In this case here, we're going back to this assay where we've got a reported molecule that's going to bind onto a kinase, and it's going to increase the polarization. What we're going to be doing is we're going to then be looking in the middle to see if we can find drugs that will actually displace the reporter out from this active site. And we get values in the middle. So how can we tell that, is this a good assay? Is this a bad assay? How can we be sure that we get a measurement in the middle of that is going to be something that's actually valid? Well, one way of doing it is we need to kind of take about what the maximum signal is in blue. What the minimum signal here is green. Look at the spread of both the values here, the maximum and the minimum. And then if we take this measure here, which is called the Z prime, which is then just a ratio of the difference of the standard deviation of the signal of the max and the minimum signal over the total signal window there of the maximum value minus minimum value. So that, as you can see here, as we were developing the assay, we can see that the Z prime was increasing up above this nice line here, 0.5. So we could be, very confident that the assay was suitable for looking then at lots of drug binding screen, across a whole, whole variety of a library. We're able to pick hits out of that. There's other ways that you can think about, your performance of your assay. You'll hear this quite a lot in papers that do high throughput screening, where you've got a coefficient of variation. You want to look at your signal to noise, signal to background, what your signal window actually is, and as we said, Z prime, and the minimum significant ratio. So if you have a look at this paper down at the bottom, there's more details about that. Okay, so you've done your experiment, and the big question is, can you repeat it? So have you actually documented what exactly you've done? Not only for yourself, but actually when you complete your thesis and the next PhD student comes along, you're long gone, and they want to be able to kind of carry on your work, can they do it again? Have you put everything in there? So a good thing to think about is you need clean enzymes. And this then quote from F.M. Racker and Arthur Kornberg: "Don't waste clean thinking on dirty enzymes." Now here's an example I did myself, where I was looking at this co-chaperone of HSP90, it's an ATPase, looking at the activity. So there's lots of these co-chaperones. We didn't really know what they did at that time. And so it was really exciting that I could, when I put this FKBP59 protein into HSP90, it massively stimulated the ATPase. And that was, this is very, very exciting. But then I did some purification just to check. And you can see there on the left, it's quite old school, we've got a chart there. So when I purified this by ion exchange, then I got two peaks. And there's a main peak here, which is, you can see the protein on the gel, you can see that. But then there's a second peak coming out at fraction 39. Very, very little protein, could hardly see it. But when I put it into the assay, 39 in blue here, by itself had a massive ATPase activity. So therefore, yeah, really what was happening was that I had a contaminating ATPase. So of course, how can you do quality control? Well, thankfully here at the LMB, we've got lots of resources that we can actually look. So the first thing, of course, is that you want to do the purity. A lot of the times people will actually do SDSPH. Have you loaded enough on the sample to see everything that's in there? Is it homogeneous? Can you check for aggregation? Well, have a look at all these different techniques here, like SecMals, which is an example of the graph down there. We've got AUC, DLS, mass spectrometry. even just looking at the UV spectrum and seeing, what the curve looks like, and seeing if you've got any light scattering at 320 nanometers. The identity integrity, have you actually cloned and purified the right thing? Well, of course, just go and get a mass spec. Is it in the right conformation? Is it stable? This kind of term of that protein being happy. And of course, we can look at the Prometheus. So that's a differential scattering ferrometry, NMR, CD. And even simple NMR here, we can kind of see from these two plots here, you can just tell whether a protein is actually folded just by the dispersion of peaks, especially down here in the between 6 and 10 ppms. How much of your protein is actually active? Do you have an activity assay? From SPR, then the relative amount of resonance units that binds onto a sample that's on the chip. We can tell what the stoichiometry is. And ITC itself, because it's a stoichiometric titration, you'll see how many active sites you've actually got. Of course, we all don't use everything fresh. We store things at various different temperatures. And again, you can check whether your protein has actually survived the process, and you may have to go back and optimize the storage conditions or the buffer that's actually originally in. So I mentioned earlier on the background signal. Do you check for that? And here's another example of a measurement. This is ITC, where it did, and this was using then a small molecule. It was in DMSO. What we can actually see here is just basically a mismatch between the DMSO concentration in the well against what was being titrated in. And it gave us lots of lovely heat here, and we could actually fit that to a binding curve, but it was just background. So other ways, you could, the titratant could give to signals like light scattering. And in this case here, it's just heat of dilution. You could have a buffer mismatch, say in AUC, DMSO and say SPR as well. You have to account for that, but luckily with SPR, then there's a program that can actually calculate the changes due to mismatches in DMSO and then adjust your values. And sometimes then the titrating has a direct effect. So say something like you are looking at fluorescence of a protein, and then you're titrating in a nucleotide. So that's going to absorb in the UV. So that's going to affect the incident light coming into the protein sample, and then it's going to affect how much fluorescence comes off. Again, there's ways to correct for that, but always be aware that that could be an issue. So you've got nice data, you've corrected your data, but have you actually got a good enough range and time? Here's an example of a published study that which shows how you can really be misled. So this is looking at small molecule screen, looking at SPR. And if you have a look at some of these binding curves, some of them look as if they are kind of, a nice kind of binding. Other ones look pretty kind of straight, as if you could put a straight line through them. So, but they fitted them and they give values here for the KD. And what they said was, well, the shape of the binding curve, well, we could just adjust that by saying if we define what the R max is, so and then the KD can just by the, it's just could be fitted as well. And we can do this in a reliable way. But, is that correct? Or is this just something that's just aberrant and it's not ever going to go up to the maximum because it's just aggregating compounds or something? Another way we can confuse ourselves is if we haven't really got the right range. And this, in case, is a titration of binding. And it's always good if you can to then plot your data in the log format with log of the concentration. And so you should see if you've got the right range is you've got this nice kind of sigmoidal curve where you've got a baseline where there's very, very little binding, then the titration step, and then you've got saturation at the top. So what can happen then if you don't have the right range? We'll say here in this case, imagine that we haven't taken enough points. Well, just because of the scatter, you can get a binding curve. So then when we go a log plot, we can actually see, well, we haven't actually reached saturation. And the KD that we're going to fit here is actually larger than the top concentration of the titrated protein. So that's a big warning sign. If you ever see that, you kind of know that, right, you haven't reached saturation. You can't really be confident in actually any of the fitting at all. So say if we then take just, not a big enough range of sample, so just at this early point, again, scatter, you could end up, fitting that looks to saturation. And in this case, then we've got a very, very low amplitude, and also then the KD looks very, very tight. So the rule of thumb would be then is the final concentration of your protein then should be between 5 and 20 times the KD. And obviously as well, you should be able to go way below the KD as well to be able to get that baseline value. One thing people can do to actually pull the wool over their own eyes is by normalizing. So in this case where we've got too few points, they've gone, okay, I think I've reached saturation, I'm just going to normalize. And so you see this fraction bound, 1 to, and it looks like a nice fit. It looks easy. But then if you compare that normalized value there to the actual real data set, you can actually see how badly you've got it wrong. So always plot raw data, don't normalize. And if you see normalized data in papers, always think, what is going on here? Another way could be is that you haven't let things accrue a bit enough. Obviously, you think in a binding reaction, there's going to be forward and reverse reactions. The forward reaction is going to then depend on the concentration of the components. And the back dissociation then may be very slow. So you kind of should do a binding curve. If you've got it in a plate format, is read it initially after 30 minutes, an hour, and even overnight, and see if there's any shift. So here's an extreme example where, as you can see, that with this reaction, basically as time went on, the amplitude increased and the KD went down. If we're looking at kinetics, have you got the right range? Have you followed the reaction for long enough? So here, typically, the full range you would want to be is 20 times the half-life here. And you can fit this exponential. But has the endpoint been reached? So imagine again, if we just take this data, replot it, and we can say, okay, we can fit this data, okay. But then if we actually follow it out to the full time course here, we can see that we've really kind of got the wrong amplitude completely, and therefore we're going to have the wrong rate. So the rule of thumb would be you need to leave kinetics at least 10 to 20 times the half-life of this reaction. Of course, when you're doing enzyme kinetics, it's the other way around, because if you want to do Michaelis-Menten kinetics on this, you're going to make the assumption that you're going to get no product formed. It's going to be very, very small, and that you're using it in a format where the enzyme concentration is way below the substrate concentration. So essentially, the substrate concentration that you put in is going to be equal to the free substrate. So then you need to then look at the initial rates. And from those initial rates, then you're going to plot that against the concentration of the substrate to be able to then fit a nice Michaelis-Menten. We also need appropriate concentrations when you're doing different types of binding assay. So here in say, FP and fluorescence or MST, we've got a label component, and that tends to be much below the KD. And that's what then again, we're assuming that when we titrate in the concentration of the protein, or in this case, then a non-hydrolyzable nucleotide, that concentration doesn't change, because the concentration of the complex is very, very small. Now, so in cases, if you get a KD that is about half the total volume of the labeled component, then alarm bells should ring, and you should kind of go, well, maybe everything is much tighter, because in this case, then what you would be doing is just doing a stoichiometric titration. So then go back, have lower concentrations of this labeled component, and do it again, and see if that you get a different result. Of course, for ITC, then it's a stoichiometric titration. So in that case, you need the protein, say, in the cell to be much above the KD, between 5 and 10. So that then when you put your other component in, it's going to be titrated in. That's going to be titrating, and so you're going to then deplete the amount of protein that's free in there. So we're going to then model this and bind it. We're going to then fit this using a molar ratio. And in the middle, then we should be able to find what the stoichiometry of the reaction is. Okay, so now we've got good data, and we we're ready to fit. So we're going to fit then with this non-least squares fitting. I'm going to have to ask what the quality of the fit is, what our estimates are, and what the error analysis is like. So here's a data model. We've got a protein then being bound by a ligand to form a complex with a certain KD. We're going to make, in this case, it's going to be, it could be anything like polarization or fluorescence. And so in this case, then we're going to make the assumption here that the formation of the complex concentration is proportional to the signal change. Here's the data model. And so we can formulate then in terms of y, which is our measured value. It's going to be some kind of function of the concentration of x times the parameters. And so y is a dependent variable. We assume there's going to be some random noise in there, and so the random variability should be Gaussian, and the percent variability shouldn't really change across the reaction. X, of course, is the independent variable. That's say the protein or the ligand that you're going to titrate in. You want that to be accurate, and you want it to be uniformly distributed across your titration range. And then this beta is the parameters. And how the computer is then going to fit is it's going to look at these residuals, which is the difference between the value of the measure, the yi, minus the function. And it's then going to sum all the square of this residual values. And it's going to go around and kind of change the parameters to try and minimize this S value. And it will do it again and again and again until it gets a minimum. So here's an example here. Binding curve, we've got some estimates, round 1, and it goes round 2, round 3, round 4, round 5, might be even up to 1,000 rounds, where it'll then kind of get a minimized to give you the values. So essentially what it's doing is you kind of think if there's only one parameter that you were changing, it's finding this minimum that's kind of going from, even if it kind of starts away from the true value, then it will kind of start rolling down this parabola and try and get down to the minimum. If you imagine then there's two parameters, then the space is like two-dimensional here, and this kind of heat map. You can see then the center is where it wants to try and go. But often, of course, then things are multi-dimensional. So you can imagine a 3D error plot here, and you've got this kind of energy space. But often there's even more parameters usually, and so you've got kind of something like a whole mountain range, and then obviously more local minimum. And so what you hope then is that you start somewhere with good estimates, and you'll get down to the global minimum. But sometimes this can happen where I'll do a search and it'll end up in kind of a local minimum where really it can't get out of, and you think, and so it will stop, but it gives you the wrong values. And we'll see in a minute kind of how we kind of judge if whatever that values that it gets, it's good or bad. Right. So what kind of models do we have? Well, in this case, this is a ligand depletion model. And this is the equation here to fit this data. This is polarization data. And if we're using a program like PRISM, we have our data, our protein concentration in X, and then we have the multiple values here in column format here in A, and they're going to average everything. We just pick things and say, right, a nonlinear regression, let's do a curve fit. And then we can choose an equation. And what's nice about PRISM is that you can actually then put these parameters into words. And as you can see here, we've got A is equal to something, B is equal to the ligand, C is equal to that. And we've got things like top and bottom, which is the starting value and the saturation value of the polarization. And then we've got this quadratic equation here with the values here. So it makes it much simpler to kind of to do out. We can then define, where the starting value should be. So, the ligand is we're going to put that value in because we know what the concentration of the label value is. The binding constant, well, it's going to be in the middle somewhere of Y mid. And then obviously the bottom and the top fluorescence polarization are going to be the Y win and the YX. And so then we kind of look at the initial values and make sure that they're okay. And then we can just fit. And then what we get out is not only our values at the top here, but if we can also get standard errors of those parameters. And also this confidence interval at 95% of what these values are. So obviously, if you look here, and you can see, well, okay, I've got this value. How much can that be varied, so that I really get I'm a good fit. And obviously, if we're getting something that's quite narrow, if you can say here at bottom, it's been 115 to 116, 0.8, and the value it gives is 116. So that's kind of very narrow confidence intervals. And so we know that we should getting an accurate value there. Another kind of way of looking at the goodness of the fit, obviously you can visualize it, and it's this R squared value here, and that's 0.9876, and that's a good value, and we'll see how that's calculated in just a second. So another value, and you'll see in other programs, is called, and mentioned as well, is this thing called chi-squared. And that is a sum of then the residuals here. You can see yi minus the function, the fitted function, that's squared. And that's then, it could be divided by this weighting function. Obviously, if there's just random variation in your y across all the concentration, you shouldn't really put any weighting in there. But sometimes you need to invoke that because as the concentration goes up, you're getting some variability in the signal. And so if we put the weighting in there, then that's going to be it's going to be a value of the fraction of y predicted from the standard deviation of y at that point. So obviously you need multiple values. And so what the R is, and it's the square root then of the 1 minus this chi-squared, then divided by the sum of then the weighting factor times yi minus the average y. So what does this bottom function means? Well, what would be the average y? So that would be taking all the y values across all the concentrations and just assuming that they're all the same value. So you count them up and you're saying, well, that's just the average. And so then you're taking away what then the difference is of the actual measured value from then the average value. And so it's then an idea of, well, how much does fitting this with an equation account for what we see over what would be just random kind of noise. And obviously the highest you get, one would be perfect, and as it goes down towards 0, then you're getting less and less, good fitting. And obviously sometimes that's very, very obvious. So the questions you have to ask yourself is really, if you look at the standard error for individual parameters, how does that compare as a fraction of the actual value that's stated? You know, is this parameter, you know, well constrained? You know, if you've got big confidence, you know, limits, then, you know, it means that the value could then vary quite a lot without you kind of changing the overall, you know, fit in terms of R squared. You know, if your error is very, very high, you know, do you have enough data? You know, do you need to get better data? You know, is the actual model correct? Do you need to then invoke, you know, a more complicated equation to try and fit the data? In the end, you know, is it physically relevant? You know, do any of these kind of values that you put into are they, you know, significant values? Or maybe you have to kind of reduce things down and say, well, let's go for a simpler fit. And a really key point here is, okay, the number of significant figures that you see here are the fitting calculations. It's in no way the accuracy of your data or, you know, the variation in a binding constant. It's just what the computer has then spat out when it's actually fitted. So never quote that in terms of, say, like what your KD is, plus and minus, you know, what it says here. So again, you know, another good thing to do is, of course, as I said before, is, you know, you can physically expect the data, look at a long view against the protein. You know, does it look as if it's a good fitting? And the more you do, the more you'll be able to kind of look at that and say, oh yeah, that's a good fit. So another way to be confident about whether you've actually got the right model is to look at the residuals. And in PRISM here, if you go to the diagnostics tab, you can then say, can you plot residuals across the X values here? In this case, we're looking at binding kinetics. And if we fit to a single exponential, then we can see there's kind of a wavy residual pattern. So you would say that there'd be a systematic error across here. If we fit with a drift term, where the signal's just drifting, oh, well, that seems to be a bit flatter, a bit smoother. Well, maybe it's a double exponential. And if we fit that, then we can kind of see, well, we're back to a bit more wavy. So in this case, we could then say that the middle one, the single plus drift, was a much better way to actually fit. So always look for systematic deviation and then decide which model fits the data best. You know, so in some cases, then you have to change the physical model. Of course, the bad thing is you don't want to invoke a more complicated equation just because you've got bad data. Now, here's an example here. We've got big ugly feet, and we've only got, you know, we've only got what, 1, 2, 3, 4, 5 data points. Well, of course, you know, it's, you know, data points, but if we put another term in here, we can actually get a nice fit. Well, the question you have to ask yourselves is, well, how can I improve that? And here's an example, another published example, where the data doesn't actually, isn't fitted very, very well at all by the model. And this is octet data. And you can see really that the fits are just not fitting at all. But still, I mean, they went on to quote their K-on and their K-off with a large number of significant figures and give us the KD. And the R squared, I don't really believe that at all. So just, you know, you don't end up like this person down here in the middle, looking like an idiot, putting a nice mathematical model on really terrible data. Okay, so let's think about outliers and outlying. So sometimes you get outliers. We can see down here at the bottom, they're kind of far away from where everything else is that you measure. Sometimes you have to remove them. In the case here in ITC, we always do an initial very, very small injection. That's because the titration needle is in the cell. And before you start the first injection, you may just get a bit of diffusion between the bulk sample and the sample that's actually in the titrating needle. So we always just dispense that with a small volume, which gives us a very, very inaccurate reading of what the heat is dispensed. So we always take that away, and that is justifiable. Other cases here might be, you're at the top of a titration of your ligand, you're getting a high concentration, you go, well, there's these couple of points at the end, should I get rid of those? I could just get rid of them, makes it all pretty kind of thing, so let's just ignore that. Well, I could be wrong. It could be that actually you just haven't got enough data range at all, and if you titrate much, much higher, you've actually got a second binding happening. So always be careful about that. Is it just happening randomly? You're seeing these kind of outliers, or can you kind of attribute this to self-deception, that you're not really kind of doing things correctly, you haven't got the right range? Or maybe, in this case, say, it could be, say, aggregation, the kind of outliers, maybe you need better assay conditions. So let's go back. We've got then a good quality of fit. We visualized the data, we looked at the residuals, the chi-squared, r-squares, and the parameter values, and we're quite confident to kind of get things going back. But what if things don't fit? What do you do then? It could be that you're just getting nonsense parameters. Well, how do we get over that? Well, obviously, if you haven't got the right data range, or you've changed the model and that doesn't fit, sometimes we need to constrain the fitting because it's just going off to nonsense parameters, or we can get over that by doing global fitting. So let's just talk about that in the next few minutes. So here in PRISM, we can kind of constrain values. So we can do things like, well, the dissociation constant must be greater than 0. That makes sense. You don't have a negative dissociation constant. Or in this case here, a real example where you can actually share parameters. So in the top one, we can see we can get a nice titration for the red. This is an anisotropy experiment. And in the blue, which is the non-phosphorylated, what happened is that as soon as you got up to higher concentration, the protein started to aggregate. And as you can see here in the total intensity from this fluorescence polarization assay, in blue, top concentrations, you're getting an increase in the fluorescence, and that's because it's got due to light scattering, due to aggregates. So I could then really say, well, I really have to ignore those points because I'm not going to be able to get good polarization values out of that. So if I want to kind of get an estimate for what the binding constant is for the non-phosphorylated protein, then I could then share what the saturation values are. And so then I would then get a global fitting here and get the top values shared between everything else. But it's really important that you actually, in your methodology, when you write it up and in your paper documentation, you actually state that you've done that. And so it's clear to everybody. So what about global fitting? Well, in global fitting, what you're doing is you've got a whole series of say, different concentrations in terms of maybe kinetics, and you're going to fit them to one set of parameters. The question you have to ask yourself is, why do I need to fit globally? Is there experimental variation, or is it just the model just so complex that actually it's difficult to fit, as we'll see with kinetic data, or are you trying to fill the balloon for everybody's eyes by just having bad data and fitting it globally? Always a good question to ask yourself is, how does this global fitting compare to individual fits? So here's some SPR kinetics here, which I fitted globally. And in this case, it was good because I can actually fit the individual on and off rates. And because there's flat response at the top, I can fit the equilibrium response. So I can find the KD from both the on and the off rates and the equilibrium position. So here's the fitting a simple one-to-one. And there's the residuals underneath. So if we fit then all the dissociation rates in red here at the bottom, you can see there's a spread, and it gives us a average value, and then in between, say, standard error of the mean there. if we did a globally fit, we're going to get one value, and that's in blue. And you can see, well, yeah, fitting globally kind of then pretty much represents, is very, very close to what the average value would be from this sample. In the association rates, it's a bit different. And as you can see, we've got a spread of values here, and really then the global fitting in blue is just at the edge there of the standard error of the mean at the top there. And that's probably because of these outliers here. And so in fitting this data, then your data, say maybe very, very low concentration, might not have enough amplitude. And so you're going to get, say, worse and worse fitting there kind of thing. But that, because you are then constraining everything to fit to one value, then they will push the global value to a slightly wrong value. So it's always important to kind of think about, well, are all my concentrations, data here, are they useful? If I say take out a couple of the concentrations, do I get the same values, or does it actually shift? So here's an example of where global fitting goes wrong. This gives a very, very low K off, and then consequently give a very, very tight KD. But if we then fitted, the initial rates of association to find out, we could find then the actual KD would be 1.8, which is similar to the ITC value. So just because the computer gives you a number doesn't necessarily mean it's right. Always be cautious. And then in the end, okay, you've got good data, you've got corrected data. You might kind of go around and say, well, I need to kind of change things slightly to a different data range. The important thing is, of course, to repeat. And also you can kind of think about, well, you want to do statistics. And also you want to think about, well, are these biological replicates or technical replicates? So here we go with KD value analysis by doing anisotropy, just lots of different days, different samples. And so the real variation that we're going to get in the KD is from doing it multiple times. So that when we put in a value of the standard error of the mean or the standard deviation, we're doing that from an average of different measurements. But you should ask, well, why can't I just average all the data together and fit that? Well, that's not a good way of doing it. You're just going to get a huge spread of data, and the assumptions say that that your bottom and your top values are always going to be the same. Just it means that you're going to be not getting a good value as well. So what about biological and technical replicates? Journals will now ask you how many replicates you've done, whether they're technical or biological. Okay, so technical replicates are quite easy. You've just done this multiple times, and in this case here, we've got the control plates from this high throughput screen, and we've done it multiple times, and we've averaged these are all average values. Now, biological replicates, these are just different protein preps, and in this case, you can actually see that as I got better at purifying the protein, then we got better and better data. So to talk about statistics, it would take a whole lecture, but you can get things like PRISM to then give you statistics. So you've got your values in columns, and then you can just do descriptive statistics, to give you the mean, the standard deviation, and the standard error of the mean. You can give you confidence limits, et cetera, and also you kind of go on and compare whether the differences are significant, but that would be for another lecture. Of course, there's other advanced things you can kind of do to kind of overcome bias. Some points in the sample could be constraining the fit. Especially if the x-axis isn't uniform. This was a case when people had to kind of do linearization in the old days, like doing the Lineweaver-Burke plot. And so that, you know, could influence what you would get out. You can kind of do advanced methods then to see, you know, whether these reported points are kind of changing the value. So this is done by statistical resampling, you know, it's bootstrapping or jackknifing, and there's, you know, regress plus for Mac and various other programs as well. If, say, you're looking at, you know, different components and you need to then multiply or add them, then you need to then look at the error propagation. It's a very, very simple formula here. If your parameter is say then additive, then really, you know, the difference there is just then the square root of the sum of the squares of the individual errors. If you have to multiply or divide your parameters, then you need to look at the fractional error over the value. And again, that is going to be a sum of the squares of the fractional values of the individual parameters. You're going to take the square root of that, and then to this you get a fractional error, then to get the deviation, then you need to multiply that result then by the total value. And in the end, it's always good to kind of compare different techniques. You know, different physical, you know, signals are, you know, involved in, say, different techniques. So, and with all their inherent biases and issues. So, you know, do you get the same answer if you try different techniques? You know, do the signals actually represent this biological process? You know, is the physical, you know, model correct? In this case here, you know, I compared ITC binding against fluorescence, and it was pretty much the same. So that was good. And of course, just repeat and repeat and repeat. So then you're going to get lovely fitted data that's going to give you a physical model, and then you can then interpret what that means about the biological model. So I'll leave you with this last thought, which is Occam's razor, which is just always go for the simplest explanation that you can. The converse of that would be Occam's broom. This is a term coined by Sidney Brenner, which was, well, you know, there's all this stuff that's kind of, you kind of sweep underneath the carpet that you kind of, oh, you know, it's not quite working, or there's kind of some kind of outliers here, and this, it doesn't quite work, and, you know, it doesn't kind of, but we'll just ignore that kind of thing. And then what he says is that, well, actually, all those kind of things don't kind of slightly work, and the outliers, maybe we should actually lift up the carpet and actually see what's going wrong and why it's going wrong, and that can give you an insight. Okay, so thanks for coming today, and thanks for everybody kind of coming to the series, and I hope it's been very beneficial to your PhD.